This tutorial gives an overview of the main features of results tables. Any template search or custom query builder search will give you a results table. Results tables are also used for list content on list analysis pages. The first thing to note on results tables is the number of rows returned by your query. The number of rows shown per page can be changed, and for large results sets you can navigate through your results either one page at a time or 5 pages at a time, or you can go straight to the last page. Alternatively, you can specify a specific page number. The second thing to note is the column headings. These give the field name for the data shown and in some cases the class that the data is from. For example, for this results table, the first 2 columns give a gene secondary identifier and a gene symbol. The next column gives the pathway identifier and the pathway name. Mousing over the column heading gives the full path that was taken through the data model to the data being shown. At the top of each column header are a number of icons. These provide the following features: column sorting, where you can set the column on which your data is sorted; remove column, which will permanently remove the column from your results; toggle column, which will temporarily remove the column; and filter column, which allows you to filter on any values that are in that particular column. And finally, the column summary, which provides a summary of all the data in that particular column. The type of summary shown depends on the type of data in the column. For example, this results table is showing pathways associated with a list of fly genes. The summary for the pathway names column therefore shows the number of each pathway in your list. Note that your results can also be filtered through the column summary. Each of these functions for each column can also be managed for the whole results table through the Manage Column and Manage Filters options. Note that most entries in results tables are links to report pages. For example, all of these genes link through to their respective report pages. In addition, mousing over an entry provides further summary information on that object. For example, for a gene, other information such as additional identifiers and the organism are given. In addition to exploring your results with the above features, you can also create lists and download your results for further analysis. You can create a list of all objects of a particular type, for example, all the genes in your results or all the pathways. Or you can select individual items from the table. However, remember that each list can only contain one type of data, for example, genes or pathways. The download option allows you to export your results in a number of formats, including tab or comma separated, GFF3, and FASTA format if you have features with sequences in your results. The download also allows you to specify the columns and the rows you wish to export, and whether you wish to compress your results. For the bioinformaticians, you can also save the code that will generate your query so that you can run it in a script. Code is available in a number of different languages. So this has been a summary of the main features of the results tables. Please see our other tutorials for more details on many of these features.